First, we grouped together the different colors of the candies, and then we moved on to the phenotype frequencies. So we just chose any random things to compare to each other, just different colors to different colors. And we did 8 green compared to 6 blue, 8 orange compared to 4 yellow, 13 red compared to 8 orange, 8 green compared to 8 orange, and 13 red compared to 6 blue. So then we came up with the ratio, which was basically the number, and it could also be reduced. For the 8 green, 6 blue, we got 2/3. And for the possible genotypes for that, we got a uppercase G, lowercase b, which it went on to the parental genotypes, which is uppercase G, lowercase g, times two lowercase b's, and it's also Mendelian. For the 8 orange, we compared it to 4y, which the ratio was 1/2, because 4 is a half of 8, and 8 is also bigger than 4. And so for the possible genotypes, we got uppercase O, lowercase y, and for the predicted parental genotypes, we got 2 uppercase 0 O's and 2 lowercase y's, and it's also Mendelian. And so on for the next ones, they're both Mendelian, and the ratio was 13:8 and also 1:1. The 1:1, no, I'm sorry, 13:8 and 1:2. So the next one is actually a 1:1 ratio. It's 8:8, 8 green, 8 orange, and that one's co-dominant because it's a 1:1 ratio and it's different. And it would just be uppercase G, uppercase O, lowercase o. We came up with a hypothesis. If there is more of a color than another color, the color that there is more of will be a dominant trait because the higher amount overrides the smaller amount. And we did the Punnett squares, so we compared our ratio to our hypothesis, and our hypothesis went well with it because looking at our ratio, the types have a larger number and they are dominant. So if they're the same, the same number is co-dominant also. So our data does support our hypothesis. Next, you're going to do your Punnett squares. You're going to make 5 of them because there are 5 frequencies. So you're going to look at what you got for the possible genotypes, and then we're going to just start with number 1. You have the GB, so you're going to look at the parental genotypes, which is really what we're going to be doing for the Punnett squares. So you have GG and BB. So it's going to start, you're going to put capital G, because it's like it's a Punnett square, and then lowercase b, and then so you're basically going to go down like this. It's going to be capital G, lowercase b, capital G, lowercase b, capital G, lowercase b, capital G, lowercase b. And then on to the next one, you got capital O, lowercase y, capital O, lowercase y. So you're going to just basically do the same thing, OY, 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 OY. The next one, capital R, lowercase r, and then 2 lowercase o's. So it's going to be capital R, lowercase o. So that's going to be R, O, lowercase r, O, uppercase R, O, lowercase r, O. And the same for the other 2. For the question where it says, what were the genotypes of the parents? Basically, all we did was just name the 2 colors of the genotypes that came from the parents that we compared to each other. For the next question, we wrote down the predicted parental genotypes because that's the different modes, and then we did the graph.